All right, so in this video, we're going to look at gene expression in prokaryotes. When we hear gene, we think of DNA. We think of prokaryote, we think of bacteria. So with this little model here, you can see that a prokaryote is a bacteria that does not have a nucleus. That's the big difference between eukaryotes and prokaryotes. Eukaryotes, like all animals and plants and fungi and protists, they all have membrane-bound organelles, or little things inside. Now, you don't see any mitochondria here. There's no chloroplast. There's no endoplasmic reticulum. There's no Golgi apparatus, and there's no nucleus. So in a prokaryote, you have the outer plasma or cell membrane, and then you've got one long circular chromosome instead of being um, DNA in strands like in eukaryotes as well. But then, as we see here, it does have ribosomes because ribosomes are the site where proteins are made. So during gene expression, we know we have transcription and translation. And during transcription, we have messenger RNA made. But then it's not like eukaryotes where you modify that messenger RNA and take out the introns and take put caps and tails on and process the messenger RNA and send it out of the nucleus to go to a ribosome. And here the ribosomes are right next to the DNA. That messenger RNA feeds directly into the ribosomes, and you're making the proteins. You're doing translation as transcription is happening, so they're happening simultaneously. So again, we see this model of a prokaryote. All the little dots in here would be ribosomes. This is called the nucleoid region. So it's still where this super long single chromosome strand of DNA exists, but they also have these little plasmids, these other little pieces of DNA that are separate from the one big chromosome. But one thing to think about is that protein synthesis is a response to stimuli. We need to turn on the process, the genetic process. So if we think about all of our cells, every single cell is not making every single gene constantly, right? We're not constantly making every protein associated with every gene in every cell. Genes need to get turned on, and genes need to get turned off as well. So this is a response to stimuli. Again, when it comes to transcription, we should be thinking DNA to RNA. And let's break down this model once, because a lot of times we're just like DNA, RNA, and we don't think that there has to be something else involved here. So again, DNA is double-stranded, RNA is single-stranded, and it's built up of individual nucleotides of sugar, phosphates, and nitrogenous bases that get linked together. But if you're going to take these individual molecules and build up a complex molecule, you need enzymes because that's a chemical reaction that's taking place when you hook these together. So, what is the enzyme involved in this process? RNA polymerase. It's an ACE, which makes it an enzyme, and it's making a polymer of RNA. So we are unzipping the DNA code, but then we are assembling RNA nucleotides to make messenger RNA. So again, when we're just writing down letters of the code and doing the triplet codons and all that kind of stuff and going from T to A and A to U, we don't often think about the enzyme that's involved with this. So when we talk about prokaryotes, we have to think about the individual gene structure. So we have a promoter region. Promoter is DNA. And we have the gene, the RNA coding sequence, and then we also have a part of the DNA called the terminator. And at this region right here, the promoter region, that's where RNA polymerase is going to hook on. And that enzyme hooks onto the promoter and it would do transcription all along this gene, and then RNA polymerase would unbind and get kicked off at the terminator sequence. Now, when we talk about operons like the lac operon, operon comes from operator. So in between the promoter and the genes, we also have a sequence of DNA called the operator. So again, we have the promoter where the RNA polymerase binds. We have the operator where repressor proteins can bind to, which ultimately keep genes turned off because the repressor will block the promoter. And we're going to look at that a lot in this PowerPoint. We also have regulatory genes. So regulatory genes are the genes that make the repressor proteins that keep these turned off. So, little quiz here. We've got 6, 7, and 8, which are genes. And in this picture, the genes are off. The genes are off. That means the RNA polymerase, this number 1, is not going and transcribing the genes. Why is it not transcribing the genes? It's still bound to the promoter sequence here. However, 2 is called a repressor, and the repressor is bound to the operator. So when the repressor is on the operator, the RNA polymerase cannot do transcription. When a stimuli is present, which takes the repressor off of the operator, now RNA polymerase would be able to do transcription, and that would turn the gene on officially to make the messenger RNA, which then can be translated into a protein. So if we have this whole idea here, bacteria have to break down sugar, right? And how do they break down sugar? They need enzymes to break it down. They need enzymes to help with the chemical reactions. Enzymes are proteins. Proteins are made from RNA. RNA, where does it come from? DNA. So in order to break down the sugar, the gene for the enzyme must exist and the gene must be turned on. So the sugar removes the repressor protein. That's the stimulus. So the sugar is the stimulus that removes the repressor protein from the operator so that RNA polymerase can bind to the promoter and in turn turn on the gene, do transcription to make the enzyme protein to ultimately break down the sugar for energy. Oofta. That's quite a lot. So this model is a great model and we'll play around with it in class and stuff, but we're going to break down and look at individual parts of this. And this is a lactose model, lac operon model. So again, this is all about prokaryotes and bacteria, so let's get a little bit more into that. And the lac operon involves E. coli. And E. coli, usually we think of E. coli, oh, that's really bad, right? However, it does live symbiotically with us. We help them to live and they help us live. So they are important for digestion. They are considered good bacteria within our intestinal tract. And they also, a lot of the good bacteria in our intestinal tract also make vitamins. So E. coli is a type of bacteria. Some types are harmless and live in your intestines. Other types can make you sick. Now, let's think about lactose. Lactose, you might have heard of people being lactose intolerant. What that means is that they don't digest lactose. So what the heck is lactose? Well, it ends in an ose, which makes it a sugar. And it is actually a disaccharide. So it's two sugars put together. As we drink milk, we have the enzyme lactase, which breaks down lactose into glucose and galactose. Now, glucose and galactose are small enough to actually be absorbed through our digestive tract and then be moved to our cells. But again, lactose is a disaccharide. Another model here, lactose is a disaccharide. It binds to lactase. Aces are enzymes. So the lactase can help do the chemical reaction to break apart lactose into glucose and galactose. So, is this picture a eukaryote or a prokaryote? We are eukaryotes, but E. coli are prokaryotes that live in our digestive tract and can also break down lactose as an energy source. But bacteria transcribe the lactase gene and translate the lactase protein, but only when lactose is present. So bacteria are not constantly cranking out lactase enzyme. They're only going to make lactase when lactose is around. So they may have a gene to make the enzyme lactase. That gene is going to be found in that nucleoid region of the major DNA chromosome within the bacteria. We have a promoter region, an operator region, and we have the gene. The promoter region is where the RNA polymerase binds. The operator region is where the repressor binds. And the gene is the section of DNA where we will get the RNA to make the lactase protein. So in this case, you would not do transcription because the RNA polymerase would get blocked by the repressor. So in this case, it would not make lactase. The prokaryote would not make lactase. But this is lactose. When lactose is present, it binds to the repressor, removes the repressor from the operator, and RNA polymerase can bind to the promoter and then go ahead and transcribe the messenger RNA. That messenger RNA can be translated into lactase. And what does lactase do? It binds to lactose and helps with the chemical reaction to make glucose and galactose. So the lactose is getting broken down when lactase is present. So all the lactase that is made breaks down all of the lactose, and when there's no lactose present, what happens? The repressor binds back to the promoter. So the transcription of lactase is stopped until you get more lactose. That's pretty complicated, but again, there's a stimuli, you get a response. To confuse you a little bit more, again, we think about the stimuli being present, which removes the repressor, but sometimes the stimuli can cause it to be turned off. So we have an inactive repressor that will not bind to the operator. It only binds to the operator when the stimuli is present. So when the stimuli binds to the repressor, now it becomes active, and now it'll bind to the operator and stop RNA polymerase from transcribing the gene. Again, so much of this comes back to being comfortable looking at scientific models and breaking down what's there. But certain things have to be in your vocabulary, like RNA polymerase, like transcription, like messenger RNA, like genes, promoters, operators, repressors. All of these words have to be functional in your vocabulary, and you have to be able to apply them to models. So far we've talked about transcription and we've kind of hit translation um, pretty quickly. But as we're doing transcription, again, I mentioned before, we have this termination site. So as it's making these genes, the RNA is being made, but once it hits the termination site, then we get this like rebinding of these nucleotides, which then causes it to kind of get kicked off of the DNA. Now, translation in bacteria It's different than in eukaryotes because in eukaryotes our ribosomes can be free floating, but our ribosomes are going to be outside the nucleus, or our ribosomes are going to be bound to the endoplasmic reticulum, and those bound ribosomes end up creating the proteins that get trapped within vesicles which go through the Golgi apparatus. But in bacteria, when you look at this model, you have the ribosome directly right next to the DNA. Our ribosomes are not next to our DNA. Our ribosomes are outside of the nucleus. So by having it right here, there can be no processing of the messenger RNA in prokaryotes. So transcription and translation are happening at the exact same time. Once it's made, we also have to fold it up, right? Because the amino acids get linked in a straight line as its primary structure. However, there's chaperones and chaperonin that aid in the folding up of the DNA of the protein of the amino acids, so that ultimately, when we have our final protein, it's a 3D shape. It's when we get a protein, it's not a straight line. All the amino acids are attracted to each other, and we end up with a three-dimensional shape because of the chemistry involved in the attraction between the amino acids. That protein then carries out functions, and we know all the different kinds of proteins that can exist and all the different functions that can exist. Again, um, this is similar. We have these in eukaryotes, but chaperonins also exist in prokaryotes. So you can take your polypeptide, all your amino acids, it goes into this big protein, which then again assists and folds it up correctly so that now it's functional. This is not functional. This is functional. And ultimately, the protein becomes functional, and then it can carry out all of the responses that are needed by the prokaryote. And that is all.